This is a video about the GCSE Biology topic of genetic engineering, or genetic modification as it's sometimes called. This comes up in the second paper of AQA GCSE Biology or Combined Science in the unit about variation and evolution. In this video we're going to identify and define some keywords that are relevant to this topic that you should already know, and then use these to define genetic engineering. We'll explain why genetic engineering may be desirable and give some examples of genetically modified organisms. We will describe the process of genetic engineering and specifically the use of GM technology to make human insulin, and then finally we'll evaluate whether or not genetic engineering is a good thing. In order to explain what genetic engineering is and how it happens, we need to be confident using quite a lot of other biological vocabulary. So we're going to quickly recap some of these key terms here. An enzyme is a biological catalyst made out of protein. So catalysts are there to speed up rates of reaction. You've most commonly met these in biology when talking about the digestive system, but actually you have thousands of different enzymes that do thousands of different jobs, and they're all trying to speed up chemical reactions. A gene is a small piece of DNA or a small section of a chromosome that tells an organism how to make one particular protein. Genome is the term that we use to refer to all of the genes that an organism has, and the genotype is which alleles an organism has for one specific gene. An organism can be any living thing, so a plant or an animal or a fungus or a bacterium. And the phenotype is the expressed or observable characteristic. So it comes about as a result of the genotype, but it's what we can actually see and what selection can actually act on. And finally, a plasmid is a small circular piece of DNA that some bacteria carry, and it can be used as a vector. So a vector is basically a way of getting genes into something else. So we use plasmids as vectors, but we also use viruses. Now that we have all those definitions, we can say that genetic engineering is when a gene from one organism is transferred to the genome of another organism. And realistically, we're talking about doing this between species, because if it wasn't between species, there are other far easier and cheaper ways like selective breeding that we could use. When this gene is transferred, it will change the phenotype of the second organism and give it the ability to make one new protein. Remember, that gene that we've transferred codes for a single protein, so it's only going to give the organism that one characteristic. It's not like Jurassic Park 3 where they accidentally made a camouflaging dinosaur using cuttlefish DNA. That just can't happen in the real world. The organism that's been made is said to have been genetically engineered or genetically modified, and we now might call it a GMO, a genetically modified organism. Many organisms have been genetically modified for the benefit of humans, either to eat them directly or so that they can produce a useful product. Although in the UK and the rest of Europe, the regulations around GM crops are incredibly tight. There are only a handful of crops that can be grown, and these aren't for human consumption yet. So there are a number of different traits that crops are commonly genetically modified to have. The obvious one is disease or pest resistant crops. So we talked before about how you might use selective breeding to breed in resistance that was already in a population. But genetic modification means that where we have one species that's resistant to a disease, we can actually put that resistance into a completely different species. Herbicide-resistant crops are major money, particularly in America. Over there, pretty much all of the maize and soy is GM unless it's specifically labelled as not. It's been modified to be resistant to a herbicide called glyphosate, or its trade name is Roundup. And this means that rather than going around and spraying the herbicide on just the weeds, the farmers can use tractors or even aeroplanes to spray the entire field in one go, and their crop won't be affected, but all of the weeds will die, and this obviously saves them a lot of time and saves them a lot of money. We can also genetically modify crops so that they have bigger fruits or that they have a higher yield, so that they're frost resistant, so that crops have an improved shelf life. Vitamin enrichment, if you haven't already heard of it, look up golden rice. So basically in India, the really, really poor people just have a diet of basically entirely rice. They don't have access to a lot of meat and vegetables. And so a lot of people suffered from a disease where they didn't have enough vitamin A, and this leads to blindness. So basically what some scientists did was to genetically modify rice with 3 genes, which allow it to make a precursor to vitamin A called beta-carotene. And you tend to find a lot of beta-carotene in red vegetables, so like carrots and peppers, and actually they used daffodils for the gene that they took. And so it means that this golden rice is kind of an orangey coloured rice, but by eating just rice, people can get enough vitamin A that they're not going blind. And then the last example we've got here is bacteria producing human insulin, which is a really big deal, and we're going to look at in a little bit more detail. Now we're going to look at the process of genetic engineering using human insulin production as an example. Often when people talk about GM, they're thinking of crops that we could eat, but actually pretty much all molecular biology involves some kind of GM technology because it's the fastest way to make proteins like enzymes and hormones and even the antibodies that make up pregnancy tests. Basically, by giving bacteria a piece of DNA, you can force them to make a huge quantity of one particular protein. And this is exactly how we make human insulin now. Previously, diabetics had to take shots of insulin from other animals which didn't work as well as human insulin, so this is a much better method. Firstly, we need to isolate the gene, which just means cut it out. And for this we use a type of enzyme called a restriction enzyme, which cuts the DNA at a particular sequence. If we use the same enzyme to cut it out of the original organism and to cut our vector, then the two are kind of sticky ends that match each other and will fit together. The small section of DNA is put into some kind of vector. Here we're trying to modify bacteria, so we'd use a plasmid. Once we've got that recombinant plasmid, so that loop of DNA that has our genes stitched into it, then this plasmid is transferred into the bacteria. And when the bacteria start multiplying and start sort of producing all their proteins as normal, they'll also produce this insulin protein. So that's how we can genetically modify bacteria, because they will take up a plasmid if you give them a heat shock or an electric shock or treat them with certain chemicals. Now, a plant won't take up a plasmid because that's just not the form that their DNA is in, so it's no good using a plasmid as a vector if you're trying to transform a plant. What we do instead is a very similar method, but instead of putting the DNA into a plasmid, we put it into a virus which can then go on and infect the plant. We need to do this at a very early stage, so when we've just got a couple of cells, because otherwise the new DNA would be in one part of the plant but not the rest of the plant. So we do this when we've got a few cells, and then we can grow them on some growth medium until they make plantlets, and then plant these out in damp soil and they'll grow normally. As with all these technologies, you need to be able to evaluate whether it's a good thing or a bad thing. So in terms of advantages, firstly, GM technology is going to be faster than selective breeding. It's not going to take multiple generations. We're just taking a gene and putting it in the organism. The second thing is that we know exactly which genes are being transferred. So this goes back to my point about how Jurassic Park 3 with the cuttlefish DNA can't actually happen. If you've taken the DNA, the gene that makes protein X, all your new organism is going to be able to do that it couldn't do before is make protein X. And you can contrast this with selective breeding, where you're taking two parents that might both have great traits, but you're mixing together all of their alleles. And so it's possible that even though they're better at the one thing you were selecting for, they're worse at some other things. Whereas with GM technology, we're only changing one gene at a time. In terms of uses of this technology, the first thing we can do with GM technology is to introduce traits that were nowhere in a species to begin with. So if you imagine there's a disease that affects dogs, and Labradors are resistant to it. Labradors are part of the species of dogs, they're one variety, and we could use selective breeding to breed that resistance into other breeds. But what if there's a disease that no dogs are resistant to at all? Well, it's possible that the resistance to that disease might be present in a completely different species, and so we can use GM technology to transfer it. And this is exactly what we do with lots of crops that are now resistant to diseases that have been genetically modified. We take the gene from a completely different species. As we've said already, this is really important for the biotech industry, where they can use it to mass produce certain proteins like the human insulin protein. And finally, modern medical research is exploring genetic modification to overcome some inherited disorders, although this is still relatively early research and there's obviously lots of ethical considerations that need to be taken into account before it can be rolled out wholesale. In terms of disadvantages, GM technology is expensive and it has low success rates. The potential for gene transfer is also non-negotiable. So what this means is that after we've developed one GMO that is, say, resistant to a herbicide, it's possible that that plant, while it's growing in its field, could have offspring with a weed, because plants are far less good at staying within their species than animals are, and they make hybrids all the time. So if that were to happen, it's possible that this could create a kind of super weed that had the genes necessary to be herbicide resistant as well. And then how on earth would you kill that weed? Finally, the effects of eating GM crops on human health have not been fully explored, and that doesn't mean that there's an issue with it. It just means that really, until we've been eating GM crops for maybe 40 or 50 years, we can't say with any certainty that they're completely safe. Finally, let's finish up with some quick question practice. Pause the video and see if you can write down an answer to each of these three questions. Genetic engineering is the process of taking genes from one organism and transferring them to another organism, usually using a vector. Some people are worried about eating genetically modified food because we don't yet know what the long-term health effects will be. In 1986, yeast were first used to make a recombinant vaccine for human hepatitis B, and in this process, enzymes are used to isolate the genes from the hepatitis B virus, and these are then put into the yeast cells. This is one of my favourite examples of really innovative GM technology. There's a soil bacterium called Bacillus thuringiensis, and it makes a protein called cryprotein, which is really toxic to insects. Basically, it makes their intestines super permeable, so everything just kind of falls out and they die a really horrible death. If maize plants are genetically modified with this gene, then they can make the same protein, and then any insect that tries to eat them will suffer the same fate. In this question, you need to describe how the gene can be transferred from the Bacillus thuringiensis bacteria to the maize plants to make what's known as Bt maize. The first step is to isolate or cut out the gene for making the cryprotein from the bacteria. In order to do this, we're going to use those restriction enzymes. Then these can be inserted into a vector. Here we're trying to transform a plant, so we're probably going to use a virus. This needs to be done at a very early stage of development, and then all of the plants are grown up and they're all able to make the cryprotein. Finally, we're going to evaluate whether it's a good thing to genetically modify the maize so that it can make the cryprotein for itself. Remember, anywhere in GCSE Science that you see a question asking you to evaluate, you need to compare both sides of the argument and then finally reach a conclusion. And it usually doesn't matter which conclusion you make, as long as you make one and back it up with evidence. So in favour of genetically engineering this maize, firstly, there's going to be higher yields because there are going to be fewer insects able to eat it. Secondly, there'll be lower spraying of pesticides. We're no longer going to need to spray on insecticide onto the outside of the plant because it's literally making its own inside it. The pesticide that it makes, this cryprotein, can't be washed away by rain, so again that's going to contribute to us needing to spray less frequently. Also, it won't harm insects that don't eat the crops. Usually when you're spraying a field with insecticides, that's also going to affect like pollinators and other insects that are in the field. It's possible, this is a disadvantage, that it could cause harm when it's eaten by humans. Also, that gene could be transferred to other species, and if that happened, then the insects wouldn't have anything to eat at all and the whole food chain could collapse. On balance, I think that genetically engineering the maize is a good thing because it only kills insects that would have died by being sprayed with insecticide anyway, and it minimises the death of other insects like pollinators. Hopefully you're now more confident about your ability to explain the process of genetic engineering and evaluate whether or not it's a good idea. Thank you very much for watching, and if you found that useful, don't forget to like and subscribe below.